bridge DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2. Each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides.
Composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification.
The total output generated in a sequencing run by the total size of the sample sequence. For example, running a human genome, which is approximately 3 billion base pairs, will yield approximately 333 times coverage on the Illumina HiSeq 2500, which has a max output of 1,000 gigabase pairs. Consult ABM's NGS experts for the appropriate coverage of your experiment here. For detailed information on the appropriate coverage required for different NGS applications, please view our knowledge base here. Prior to sequencing, the sample library must be validated quantitatively and qualitatively to verify if there's a sufficient amount of good quality DNA. What is considered a good quantity of DNA depends on the library protocol's specifications. Having either more or less DNA results in less efficient sequencing.
Indices and regions that are complementary to the flow cell oligos. Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer.
Then to position 2 molecular blades to cut each strand of the DNA double helix. Repair enzymes seal the gap in the broken DNA, sometimes by inserting new genetic information supplied by the cell or the experimenter. In this way, disease-causing mutations can be corrected by changing the underlying genetic code. Scientists are now using this Cas9 technology for research applications in human health, agriculture, and bioenergy.
Think of DNA as the instruction manual for an organism.
In people, genes can influence characteristics such as your height and the color of your eyes. DNA is inherited from our parents. We have a combination of two sets of genes, one from each parent, brought together to produce a new set of instructions and reshuffled in each generation. But sometimes these instructions carry errors, and these can create problems. Faulty genes can cause serious illnesses. Humanity has a long history of figuring out ways to modify genes. We have been crossbreeding plants to make them better to eat for thousands of years. But with gene editing, we've made a great leap forward.
Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates.
Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA. One of
we also send you a little bit of protein and instruction for a gene surgery. When Lulu and last month was just a single cell, this surgery removes the doorway through which HIV enters to infect people. A few days later, before returning Lulu and Lana to Greece water, we checked how the gene surgery went by whole genome sequencing. The result indicated that the surgery worked safely as intended. Grace's pregnancy was normal, which we monitored closely by ultrasound and blood tests. After birth, we again